biological seminar series. Today I'm pleased to introduce Alexandra, our local guest, but doing really great science. Alexandra completed his PhD at the Federal Institute of Technology at Lausanne, Switzerland, in the Laboratory of Electrochemistry about the protein separation analysis methods. And then, actually, for the graduate students, you're lucky that you're not in Germany. In German field, if you have a PhD, it doesn't make you an advanced person to be in academia and be a professor and so forth. You have to complete something more higher and extensive than PhD, that's I've been told. But I believe all these people call habituation or habituation, if I butchered in German, and Venia Legandi in 2007 in experimental physics. He is an expert in microfluidics and nanofluidics, especially for protein or nanocrystal separation, protein analysis, and biomolecular separation. And today he's going to us talk about that. Thank you.
Okay. Well, thank you very much for the kind introduction. It's always nice if people make the effort and try to look at the differences, right? And so just one comment, so this Venia Legandi thing is kind of weird since it gives you the permission to teach, or in order to get it, they ask you to teach. So it's kind of weird. I think that's really interesting. So you have to kind of talk for your advisor during that time, and then at some point you get permission to do it officially. So I'd like to start my talk with a short introduction on microfluidic platforms and microfluidic systems, which are also often called lab-on-a-chip systems. And I brought an example how an analytical chemist might look at these systems. So what you see here is schematically depicted such a realization of a microfluidic system. And in red you see microfluidic channels, and the circles here represent the reservoirs with which we can access these channels with fluids. And then most of the time, at least that's what we do in our lab, we put electrodes in these microfluidic reservoirs, and then we apply potentials, and this is how we try to manipulate biomolecules, nanoparticles, and other things in these microfluidic devices. So if you want to build a separation system, then you need a few important components. You need an injector. This is here, and then a separation channel where your analytes separate, and then at the end a detector, which can then analyze the separated parts that flew by. Something about the dimensions. So I put here that the cross-section of such a channel is typically about 100 micrometers to 100 micrometers. That is about the wingspan of the smallest insects found on Earth, these fairy flies. And here is an example of the Tinkerbell and Anna. You don't see really the wing here, but it's about 100 micrometers wide, or in other words, the thickness of a human hair. So this is typically the dimensions we talk about when we talk about microfluidic systems. So a few decades ago, analytical chemists realized that there is a lot of advantages involved in these microfluidic systems, and those include cost reduction. You know, this is always a factor. You need less sample, less chemicals. Then you can easily parallelize them. So one microfluidic system is nice. You can bring it on a typical credit-sized card or so, but you can parallelize the systems pretty easily since these separation channels are so small. So there is the potential for high throughput. Very often they're very rapid. Some show improved performance, which is nice too, and they're versatile because they're so compact. You can carry them around, and they're easy to use, but maybe my graduate students might have another opinion about that. And so these are two examples from devices that we filled with dye, so you can see those, you see the channels and the reservoirs. And then here, this is actually from a commercial device from the Agilent system. And since this is around for a while, there are a few microfluidic systems that are now commercialized and sold. This was the direction. Okay, so over the past decades, there has been a large interest in from benchtop systems, such as typically capillary electrophoresis, miniaturizing into the microchip format. And this is an example for the Agilent bioanalyzer. So the whole instrument is about a desktop computer in size, and the cartridge where actually the separation takes place is in here. It's about an inch by an inch squared, so it's really tiny, and the active area where the microfluidic channels are is about a centimeter by a centimeter squared. And then there's more engineering effort that actually went into the miniaturization and true lab on a chip where you can actually take this thing with you and carry it around, so you can hold it in your hand, and all the components that you need are actually integrated. But there's more, and so I brought 2 examples here. Since sample manipulation is really made easy, so you can manipulate volumes of nanoliters to probably femtoliters really easily, and even single molecules. And I like to show that movie, which demonstrates that you can manipulate single molecules if you wanted to do that. So we're flowing DNA. This is the light blue part from top to bottom. You've probably seen it. I can start this again. So if we do this in an adequate way, we can inject a single molecule with this method. So I'll show you when it comes. Here it was. Okay. I've seen this so often, so it's so clear to me, but believe me, you can inject. So I don't have a real application, but I just wanted to demonstrate that it's really possible to manipulate either a very low volumina or even single molecules. And then the same relates to cells. You can also easily create a microfluidic device that holds a cell and in which you can manipulate a single cell. Okay. So everything I told you so far was related to what we chemists call open tubular channels. So a capillary, either round or squared, in which we try to perform separations. But additionally, we can structure these microfluidic devices by microfabrication or nanofabrication techniques. These are 2 scanning electron microscopy pictures that show you how we can integrate posts, or we can integrate these cavities. And then with using this additional structuring, we can actually tailor the environment, and so we can exploit this for novel migration mechanisms that would not occur if we would do this in open tubular channels. And 2 examples here are biomolecular dielectrophoresis, some other applications. I will go over a few of those today, and then absolute negative mobility. And I will also show an example about that in my talk. So this is a short outline of what I've planned. I wanted to just briefly go over governing electrokinetic effects that we exploit in these structures, microfluidic devices, and then talk a little bit about dielectrophoresis with biomolecules, how we can exploit this for DNA separation, and then show our efforts on proteins. And then I think there's a large bioXFEL crowd here, so we'll also talk about exploiting DEP for nanocrystallography. And then I wanted to spend a little bit of time on this since it's brand new, and since we just figured out how we can come up with a new way for this weird paradoxical migration, absolute negative mobility. Okay, so the electrokinetic effects that we have to understand are at first electrophoresis and electroosmosis. So I think you're probably all familiar with the concept of electrophoresis. You put a charged particle in a solution, and you put it in a solution in an electric field. This particle will get accelerated as balanced by the drag force, and so it migrates at a speed which we call the electrophoretic velocity. And it's characterized by some properties which relate to the drag and the actual charge of that molecule or particle. And so we can write it up as the velocity equals to an electrophoretic mobility times the electric field. Electroosmosis is another effect that occurs specifically in capillaries, with charged channel walls. There we create an electrical double layer, and if we apply an electric field, the ions in this double layer are accelerated, and due to viscous drag, they drag the liquid with them, and so a bulk flow results. And this bulk flow has a typical flat characteristic profile. You probably all know in a pressure-driven flow, you get a typical parabolic flow profile, and electroosmosis is very characteristic. You get this flat profile, and for many separation applications, that can be advantageous. You can also describe an electroosmotic velocity, which relates to a bulk flow now. That is characterized by electroosmotic mobility, which relates to the stator potential in that system and the electric field. Okay, So to emphasize on that, those are two electrokinetic effects that occur in homogeneous electric fields. Okay, so electrophoresis, as mentioned, probably all familiar with that. You can do this in slab gels. For example, you can do it in capillaries, and you probably also know that you cannot separate DNA in a free solution, even in a gel, in order to size distinguish between different DNA lengths. And so, if these electrokinetic effects are not enough, you can add another migration mechanism, and we're looking here into dielectrophoresis. Now, the difference to the other two that I mentioned is that dielectrophoresis occurs in an electric field gradient. Okay, and so I brought this equation here with you, which describes the dielectrophoretic force, and you see that it is proportional to the radius cubic, and then this Clausius-Massotti factor, and the gradient of the electric field squared. Okay, so if we wanted to exploit dielectrophoresis, then we need to kind of create electric field gradients. So it's nice because it's size dependent, but it also depends on this Clausius-Massotti factor here. You see that is described over the permittivity of the particle, and I'm sorry, I mixed this up again here. So this is the particle, epsilon 1 here should relate to the particle permittivity, and 2 to the medium conductivity that surrounds the particle or the bimolecule. And then this reduces in the low frequency limit to the conductivities, and here again this sigma 2, which is 1 over here, is the particle, and the other number is the medium. And so then if you balance that with the drag force, we can also come up with a DEP velocity, and now we see that we can sum up the factors here up front and the DEP mobility, and now it scales with the gradient of the electric field square. So this is the difference, right? Electrophoresis and electroosmosis scale with the electric field. Okay. So what does it mean? Since it's a gradient technique, you can basically attract particles or biomolecules to regions of highest electric field gradient, or you can repulse it from them, and then we call this positive dielectrophoresis if this Clausius-Massotti factor is positive. And then we have attraction forces, whereas we call it negative dielectrophoresis. Then the Clausius-Massotti factor is negative, and then we have repulsions from the regions of highest electric fields. Okay, and so it's nice, it's a gradient technique, so you can trap if these trapping forces are large enough, and you can combine it with the other effects, with electroosmosis and electrophoresis. So the next question is, how do we generate electric field gradients in microfluidic devices? One of the first approaches was a so-called electrode-based dielectrophoresis. So you can pattern microelectrodes in your microfluidic channel, and you can then create electric field gradients, as shown here, around the electrodes. So basically, these white lines should be electric field lines, and then a gradient, del square, is shown with the color coding. You see that these gradients are very localized around the electrodes. And this method also has the disadvantage that electron reactions so interfere in your separation systems. And you can alternatively perform insulator-based dielectrophoresis. And I brought an example here of a microchannel that is made of, the entire device is made of an insulating material. And additionally to that, we put in insulating posts. Now, if you look at two posts from the top, you can actually see that these electric field lines are then squeezed through here, and then we can actually compute the electric field. This is a top view of triangular posts, and you see that the electric field here is enhanced, and it also we create a very large gradient, or del square, in these devices. So this is insulator-based DEP, and everything I'll show you will be based on this type of generating electric field gradients. Okay. Okay, so then we talk a little bit of what we can do with biomolecules in terms of dielectrophoresis. And if you remember the first slide I showed you and how we can build a separation system, we took the same approach, and now as the selectivity criterion, we put an array of posts in the separation channel. So we have a crossing of two microchannels that serves as the injector, then a separation channel, which includes an array of insulating posts, which create these electric field gradients. And then at the very end, you can install a detector or some sort of fluorescence microscopy or a point detector. in order to see, analyze the combine, or you can use also a sensitive camera to see what happens. And actually that's what is shown here. So now under a low frequency field, we look at a microfluidic channel. So the channel is actually along this axis here, and the light dots you see are DNA molecules that are stained with a fluorophore so that we can observe them with a fluorescence microscope. And now what happens is once we apply the field, what I should also say is it's very hard to see. So we have rows of posts with tiny gaps, and in between these gaps these electric gradings are created.
How long are the DNA molecules?
So this is lambda DNA, so it's 40 kilobase pairs, the micrometer size. Not in length. And so you see what happens is these molecules actually get trapped. So the dielectrophoretic force is large enough so that we can attract these molecules to the dielectrophoretic or to the regions where the highest gradient acts, and then once we release the field, they diffuse out of the traps again. And so now you can overlay this with a transport mechanism, and that is actually shown in the next movie here. So you if you overlay with the DC component, which leads to an electroosmotic flow, now we can inject a sample and we can overlay with this attraction force due to dielectrophoresis. And so you will see an injection. So we injected the plug here, and you see that some molecules are retarded. We'll stop it for a short while and then we'll continue. So this is a mixture of two different DNA sizes, and you see that some are trapped earlier and some are trapped later. So we exploit this effect of that larger molecules actually experience larger dielectrophoretic forces, and so we can actually induce a smaller migration velocity than smaller molecules which experience a smaller dielectrophoretic component and can migrate further in a separation channel. And so with that method, we can separate long DNA molecules. This is an example for T2 DNA from lambda DNA, so 164 kilobase pairs from 50. This is an example for supercoiled DNA. This is a typical dimer separation, a 7 kilobase per supercoiled DNA versus 14 kilobase per dimer. And then we also figured out that we can do topoisomer separation. So a linear DNA molecule of the same length has a different dielectrophoretic attraction force than has a coiled or a circular closed and coiled DNA molecule. And so we can use this also in order to separate. So you see here again, so this is the CCC DNA, so the supercoiled part, and when we open it and linearize it, then we can see that we have another peak horizon here, which actually relates to the linear form of DNA. So to emphasize, this is a really fast separation, but these are long DNA molecules that separate in about 2 minutes. If you do it in a slab gel, you need probably a few hours to do that. And what else? Yeah, if there would be nothing else in the channel, we would not be able to separate, right? So if there is not anything that induces the size selectivity, such as dielectrophoresis, we could not make the separation because all DNA, it doesn't matter what length, would migrate at the same speed. Okay, so then we thought we wanted to look into other DNA species and see actually whether smaller ones would also be, or if we can generate forces that are large enough to trap smaller ones, and whether we can do this with artificial DNA. So we looked into DNA origami, and we tested a 6-helix bundle DNA origami. And you see this, if you don't see it, well, I'll tell you. So this is an artificial DNA that is made out of a circular construct from which a sequence is known. And then if you hybridize it with shorter fragments, you can actually fold it in a specific form. And here this was done in a way that this bundle is created. It's about 80 nanometers long and a few nanometers in diameter. Now you can take the same template and just use other shorter fragments, and you can then fold it into a triangle. So this is a triangle origami, and this has a side length here of about 130 nanometers. Okay, so we were interested whether we can also manipulate these two DNA species with dielectrophoresis. And so a typical experiment is shown here. we're not coupling this to a separation experiment yet. So it's just looking at a structure that has microposts, and we apply a small frequency AC field, and then we'll see whether we can trap these origamis. This is how a typical experiment looks like, so it's a little bit more tricky. The intensity of these is way lower, they're much smaller, and you see, but still you see that we get these trapping effect with the origamis. So we look pretty extensively on the conditions under which this happens. And it's interesting, so that we see different frequency ranges for different applied potentials. And these are now the potentials that we apply in the electrode reservoirs. The actual electric field and the gradient that acts is in the device is pretty high. So you see that we get the higher we go in the applied potential, the higher we can go in the frequency in order to trap these species for the 6-helix bundle. And for the triangle origami, we tried for quite a while, and only at the highest potential we could apply, we can actually see some trapping effect occurring here, and the frequency range also is pretty low, only between 300 and 1500 hertz. So additionally, we saw this effect in the trapping, and what we call the trapping length. So in between the posts is the area where the largest electric field gradient acts. And so we noticed that the higher we go in the frequency, the smaller this area gets where the molecules are actually trapped. And we could actually backtrack that to an overlay with the electrophoretic motion that these molecules also have, since they are charged. And so since the molecule is actually wiggling around in these dielectrophoretic trap, and the faster you go, the less it can migrate by electrophoresis, and so this is why it actually shrinks in the trapping length. So this is where we are with the DNA origamis, and we also started questioning ourselves whether we can exploit dielectrophoresis for proteins. Now for proteins it's pretty tricky. They're much smaller than DNA, and so we would expect much higher electric field gradients in order to manipulate them in a way so that these dielectrophoretic forces become somewhat important. So we played around a little bit with the geometry, and our first attempt was these triangular posts with tips. In this simulation, actually, we never get them that sharp, but this is what you see. The idea, this is what we can realize with typical photolithography techniques. Still, these triangle structures are about an order of magnitude larger in the Delhi square values that we gave it a try. However, I wanted to show you our first attempts here, and this is shown in that movie. So you see these light spots showing up when we apply. Now this is under a DC component. When we apply this, and we figured out that these are just too large particles here, so these are protein aggregates what we trap, so it's not really protein monomers. When we added adequate molecules in order to avoid this aggregation, then we saw a very different behavior, and this is shown in this graph here, or in this image. So we have a channel here. The channel goes from top to bottom, and you see when we look at the fluorescence image, we see basically that we have these streamlines. So there are regions of higher intensity. So we do not induce a trapping effect, okay? So we're not yet trapping this proteins, but we're reducing so-called streaming dielectrophoresis. However, it was a little bit puzzling in the beginning to see what happens. So if you look at the largest electric field or Delhi square values, we would expect this to happen here. So if we would assume there's positive dielectrophoresis with a positive Clausius-Massoudi factor, we would expect the DNA to actually increase in concentration at the tips of these triangles. However, this happened over here in the regions where typically you would expect a negative DEP to occur, where these Delhi square values are the smallest. And so, since it was reported in literature that proteins, and it's basically, I forgot to mention it on the slide, we use an IgG molecule here, so that these show positive dielectrophoresis. We weren't quite sure what happens. So we did a test in terms of the medium conductivity, I showed you that the causes and side factor is dependent on the medium conductivity. So by increasing the medium conductivity, basically we saw a decrease in the DEP effect, and this is in accordance with positive DEP. The causes and side factor would become smaller when we increase the medium conductivity. So that was the first indication this should be positive, although we see this largest intensities here in the negative regions. So next we looked at how can we possibly model this, because it puzzled us a little bit. It was just not intuitive to backtrack what we saw experimentally with our understanding what happens in terms of molecule migration. So we started to do numerical simulations and basically assuming that our total flux is provided due to a diffusion component and to convection transport due to an electrokinetic velocity and a dielectrophoretic velocity. And so then we solve this at the steady state here, and we then set out to model a positive and a negative DEP case. So the values that we had for the positive DEP case were actually from literature for people that determined the protein connectivity here. So we use these parameters to come up with a DEP mobility, and then a diffusion coefficient for this particle. And then we know the electrokinetic component, or basically the electroosmotic component in our device here. So we put that in. It's not on the slide, but we also know this mobility. And then just to come up with a negative causes and side factor, we assume the particle conductivity of 0, and then we look at the differences. So here again, you see the electric field distribution and the Delic square values for this case, and then we looked at the solution, the concentration distribution actually in the streaming dielectrophoresis case. And what you see is that assuming a positive dielectrophoretic mobility, you see exactly the streamlined concentration happening. Whereas when we assume a negative DP, and this is really hard to see now, it looks like nothing happens, but actually what you see is a slight depletion in concentration at these regions that correspond to a concentration effect here. So this led us to assume that we actually deal with positive dielectrophoresis, and that in the streaming case, the concentration occurs here. And in negative DP, we would have a depletion in this area. I have another slide shows this effect better for negative DP. Okay, so we also noticed with playing around with these numerical simulations that if you reduce the electroosmotic component, you can increase this concentration effect. And so what we did is we changed our coding strategy, and we added this block copolymer into the buffer that we used for the dielectrokinetic experiments. And so the puzzling thing was that the picture now looked completely different. We now get a depletion. Actually, you see these darker areas relate to depletion. And this actually relates pretty well to a simulation for negative dielectrophoresis. And we believe what happens, and we actually checked that was the case. So we created mice cells of this block copolymer. And so our protein embedded in this mice cells actually now showed negative dielectrophoresis, whereas the protein without or not in a mice cell did show positive dielectrophoresis. So what I showed you relates to streaming dielectrophoresis, and is not good enough yet in order to make it some sort of separation experiment. So what we wanted to do is increase these electric field gradients in order to better concentrate proteins, and in order to not change the entire microfluidic environment that we know so well. All our devices are made out of PDMS, and we know pretty well how to treat the surfaces. So we decided to stay with the same approach of making the microfluidic channels, however, including some nanostructuring procedure in it. And so this is shown in this series of images here. So we make a microfluidic device as usual, which basically, Okay, as usual means that we start with a master from which we mold a polymer device. And so typically that polymer has holes, not the polymer, that master has holes in the photoresist that translate into posts in the microfluidic device. Okay, so what we need to do is create holes, tiny holes, in between these micrometer-sized holes. And the method of choice was focused ion beam milling. So we could use our existing masters, and then we would mill nanometer-sized holes in these devices. And the result is shown here. So we can then post, you see a triangular post with a tiny nanometer-sized post in between. Why did we look into that? Because we noticed that we can actually increase these Deli square values quite a bit. So this is without a nanopost. You see we have something in the order of 10 to the power of 16. And if we go into nanostructuration, we can do 2 to 3 orders of magnitude increase in the Deli square value. And actually, that worked pretty nicely in order to increase this concentration effect. It also looked a little bit different. So if we have post array, for example, and this is now the protein PSA, and you see a pretty large concentration effect. We have also then our IgG here. Also here, the interesting thing is when we do this with IgG and the nanopost, we can reduce the applied potential quite a bit, and we still get an increase by about a factor of 20 or so in the concentration. And we can also do this for beta-galactosidase, and that was the first case where we saw something different happen. So a concentration on the right side related to the flow direction. So the flow direction in each of these cases is from right to left. In these two cases, we see protein concentration on the left side of the nanopost, and then this one on the right side. And we believe that this is due to an overlay of positive dielectrophoresis with the electrokinetic component that we still have, and this here is due to a negative dielectrophoresis with the overlay electrokinetic component, so that actually we get the protein concentration on this side. Okay, so I have not shown you a separation device yet on proteins, and we still have to put a little bit more effort into that. You can imagine that this is quite a cumbersome method to actually make these nanoposts, and so we're currently changing our strategy and looking into other ways in order to obtain constrictions that are below a micrometer. And then we can actually extend over an entire microchannel that is a few centimeters long, and that's our problem. So this is a manual process where you have to actually mill each hole, and so we're looking into something that we can do through photolithography, so that we can construct a separation system. Okay, so as next I wanted to show you how we also use dielectrophoresis for nanocrystallography. And so I will just briefly introduce how or why any sort of separation or fractionation of crystals is important for crystallography. Okay, so here at ASU, you know that there is a huge collaborative group that deals with this novel crystallography technique of femtosecond crystallography with X-ray FELs, electron lasers. Why are we so interested in that? Because with that technique, we want to resolve the structures of very complex proteins. One example here is photosystem 1. Photosystem 1 is a really complex protein consisting of about 380 cofactors and about 40 different proteins. So in nanocrystallography, in serial femtosecond crystallography, what you do is use this really powerful XFEL, and you shoot with it on a stream of liquid that contains crystals. Each time you hit a crystal, you get diffraction patterns, and from those diffraction patterns, you can then determine the structure. So this laser radiation is so intense that you destroy each crystal, so this is why this technique is also called destruction, detect before destruction, right? And you put diffraction before destruction, something like that, right? Okay, so what can we do with the microfluidic device related to zero femtosecond crystallography? Well, since people have tried to grow larger crystals for the traditional crystallography technique for a while, so that the current methods to grow and to harvest nanocrystals are still in the developmental phase. For specific reasons of data analysis and also for reasons of detector damage, such that usually the beam intensity is attenuated if there are larger micrometer-sized crystals in a sample. Size homogeneous nanocrystals are also desired, and there's practically nothing on the market yet that actually can perform that. And also this the liquid jet design also requires large quantities of nanocrystals. So we thought that with these microfluidic approaches, we hopefully can contribute for sample preparation in zero femtosecond crystallography. And this is by methods of separating and fractionating membrane crystals of sizes smaller than a micrometer. And then also develop tools to optimize the conditions of crystal growth. So I'm not going to show this part down here due to time reasons today, but I want to show you how we can exploit microfluidics and dielectrophoresis for separating crystals into fractions of nanocrystals. Okay, so the idea we came up with is the following. So this is the schematics of microfluidic channels that we use for this approach. We have an inlet channel here, which leads into constriction, and then a series of outlet channels. So this is a zoom-in picture, so we have a larger inlet channels, a constriction, and then this goes into 5 different outlet channels. This is a bright field microscopy image. Now, if you apply potentials at the end of these reservoirs, we can also see that we create electric field gradients at this constriction here. And this is actually what we would like to exploit for separating crystals of different size into different outlet channels. Okay? So first we looked at the realization of the technique, where it's really helpful in order to fractionate particles. And we employed polystyrene beads of different sizes. So we looked at about 100 nanometer beads and 1 micrometer beads. And again, first we performed some numerical simulations to save us some valuable experimental time. And this is basically the same concept here. We look at the flux, again, which is given due to a diffusion component, and the convection, which relates to an electrokinetic component and the dielectrophoretic component. Now, the nice thing is with these beads, they have been extensively studied in dielectrophoretic applications, so their dielectrophoretic properties are pretty well known. So we know that they have a negative dielectrophoresis, and we can come up with a dielectrophoretic mobility for them. We can also determine the diffusion coefficients, and then again we know the electrokinetic component, which in our case is just due to electrosmotic flow, since we neglect electrophoresis in that case. And so then we can again look at the concentration distribution that we obtain in such a system. And so this shows you 2 results with these 2 different particle sizes. And here the color code means yellow is basically nothing, and black is the highest concentration, or the concentration we provide to the system. Now the interesting thing is, first of all, nothing happens if we take the smaller particles, okay? So you see, under these specific conditions that we use for the simulation, the concentration distribution is the same in all outlet channels. However, if you take a larger particle size, we see that we can concentrate the particles in the middle or in the center channel. And this comes due to the effect that the dielectrophoretic force by these particles is larger, and that they actually, because there is a negative dielectrophoretic force acting on them, they get pushed into, or they get deflected from these regions of highest electric field gradient and center, and then actually flow into the center channel. And with these simulations, you can play around, so you can take the dielectrophoretic component off, And then you basically see that nothing happens, so the larger ones will do the same thing as the smaller ones. And then you can certainly play around with the potentials you use and the conditions in order to make this sorting effect the best. So this worked pretty well. I wanted to bring out a movie, I just realized, but it doesn't matter, so it just doesn't as well. So if you look at the 1 micrometer beads, I started the movie here. Actually, you go from a low potential to a higher potential. So now comes a higher potential, and you see initially they went to all the side channels, and now the beads mainly focus on the center channel. So by increasing the potential, more of these beads end up in the center channel. And similar things happens for the 100 nanometer beads. This goes from a high potential now to a lower potential. So we start out with a pretty high dielectrophoretic component, which even makes a smaller particle center in the center channel here. And then you lower the applied potential, and then you see that eventually they migrate in the other side channels as well. So when you lower the potential, you see that this works. So then you can perform some analysis on the fluorescence intensity, and we can do tracing analysis with the larger beads that we can resolve individually. And you see that this is working out pretty well. And then we tried the same setup with nanocrystals. So that's what we're actually interested in. And that's what you see next here. So again, this is the schematics of the device and the conditions that we used in order to make this work for nanocrystals. And the movie shows you now photosystem 1 right off the crystallization broth as we get it from the group. And when we start the movie, you see that there's a really large distribution in the different sizes of crystals. And the larger ones, micrometer size, they end up in the center channel. We can see them individually. And then the smaller ones, they're so small that we mostly cannot resolve them individually, and they end up in the side channel. So what looks as background fluorescence is basically the nanocrystal. And certainly we wanted to further characterize them, so we looked at dynamic light scattering and also the size distribution just by fluorescence microscopy. You see this is the bulk solution, so pretty size, inhomogeneous. Then the focused, that's what we find in the side channels, is very, sorry, that's not what we find in the side channels, that's what we find in the center channel. Still has these very large particles in it, and then the deflected, that's what ends up in the side channels, has a pretty nice distribution, about 100 nanometers. You don't find these larger particles any longer. So this is pretty nice. So I wanted to check how much time I have left.
Yeah, sure. You have
10, okay, so that gives time to look at the A&M. Okay, so then my last part is here. Oh no, it's not yet, so only 10 minutes. Okay. So for the nanocrystallography people, let me tell you, we're looking into upscaling that. You will probably ask me, okay, what's with the flow rate of this crystal sorter, okay? So we have a microfluidic device, and the flow rate was pretty low. So that's the disadvantage of that. So we were looking into upscaling this device, and in the same way, so improve the throughput, but retaining the selectivity. And so our second generation device actually has much larger reservoirs. It has wider channels, and it has a wider constriction. But we wanted to basically keep the selectivity of this device, and maybe I will go over this a little bit fast. So we used our numerical simulation tool here to come up with criteria. When is the separation or fractionation good enough? So we define a sorting efficiency here for that, which is basically related to the ratio of the larger and the smaller species we find in the side channel. We also came up with an improved angle here. And so this is now an example of, under the same conditions, 500 nanometer particles and 2.5 micrometer particles. You see that the larger ones are centered, that's what we want. Smaller ones end up in the side channels. And so we can also find a really nice transition in when the sorting efficiency kicks in, and we look at the parameters exactly when we can match this And so we have now a really good handle on this upscale device in terms of how good it would work. And this goes even so far that we can come up with different designs. Let's say we want to separate 100 nanometers from 1 micrometer at a 50 micrometer constriction, or we want 500 from 2.5 nanometers at 150 micrometer constrictions. We can look exactly at the points where actually a good sorting efficiency is met for different inlet potentials, for example. And then we can also look at the flow rates that we observe from that. And here, this is actually where we want it to be. So with a high sorting efficiency, we now end up in the 200 microliters per hour range. So this is way, way better. So we hopefully will be able to test this in upcoming screening review time in November with this upscaled sorter. Okay, so that just very quickly. Okay, and then lastly, I brought this DP-induced absolute negative mobility. So with that, I need to explain a little bit first of what we have done previously and what absolute negative mobility is, and then I'll show you how we wanted to exploit dielectrophoresis for this kind of weird separation mechanism. So A&M relates to paradoxical migration mechanisms that occur under non-equilibrium conditions. You've probably heard about ratchets. That's a related technique. And I think it's the easiest to look phenomenologically at A&M. It's a probably prerequisites that we need in order for this mechanism to work. So we need a non-linearity in the system here, which we again induce through microstructuring, so posts in the device. This goes through an entire microchannel. We need Brownian motion for it to occur. And we use colloidal particles for that. And then we need to induce this non-equilibrium effects, which is the periodical driving and an AC potential. So similar to what we've done previously with DEP, but at a much lower frequency. And then we perturb this system with an additional force. And the weird effect seen in absolute negative mobility is that if this perturbation is negative, we see a positive velocity, and vice versa. So how can this be realized? And I'll show you in another slide how this actually looks like applied. So how we can realize it is with a system that shows a gap and a trap. So these are, again, rows of posts arranged in a specific way where we have gaps that are large enough to let all particle species through, and then a trap that is small enough so that it serves as a geometrical trap. Okay. Then we subject beads in the system to a periodic AC potential, and then we overlay it with the perturbation with a DC offset. Okay, so the characteristic, this is hard to see. So the characteristic of A&M is now that if this DC offset is positive, we end up with a negative particle velocity. So the blue dots are experimental observations, and the red line was actually simulated for our collaborators. And you see if this offset is negative, you get a positive velocity. So basically that means you can transport particles against the direction of where the largest force acts. Okay, so that's the characteristics of A&M. It can only happen because we are under non-equilibrium conditions. And then what is seen very faintly is this line of this gray line here, and that relates to the particle movement if everything would be fine. Okay, so if everything would be fine, it would have a positive offset, and my particle would follow the direction of that positive force. Whereas in A&M, it actually migrates in the opposite direction. Okay, this is why this is occurring. So we can split this periodic driving up into 2 half periods. And so in the first half period, is shown here, when actually the lower potential acts, what you see is that a particle starts migrating and actually now can migrate through a gap. It encounters a trap, and then there's a certain probability for it to overcome this trap and migrate further. If that happened, in the second half period, we have a larger potential acting, so the particle now migrates backwards, but it migrates much faster. Now, because it migrates so fast, it encounters the trap early, and so the probability to overcome this trap is lower. And so this is why, if this process happens a few times, you can transport a particle to the right, although the average force points to the left. And this was actually demonstrated a few years ago to occur with colloidal particles. Okay, so we were recently interested to separate mitochondria and do this by size. Mitochondria have some important cellular functions, and mitochondria appear in different sizes. So they appear as normal and as giant mitochondria, which can be up to 5 micrometers in size. And so it's discussed that these larger mitochondria have some important function in aging. And so we discussed this with some collaborators and came up with whether we can use this A&M effect to separate organelles as small as mitochondria by size. So the problem we have here is that the mitochondria, the smaller ones, they are too tiny in order to use them in an A&M setup. And so we wanted to come up with another method in order to trap particles. And here we use dielectrophoresis as the trapping mechanism. We did some preliminary study where we looked at whether these mitochondria actually can be trapped by dielectrophoresis, and yes, we found out they show negative dielectrophoresis and low conductivity and also low frequency. And so this is why we based a model of A&M on negative dielectrophoresis of these particles. Okay, so the question was, can we adapt this effect or this geometry and then induce A&M with smaller particles? And so the idea is here, this is just a section out of a microfluidic device. We have these rectangular posts down here. The electric field is applied from top to bottom. And then you again see that we create these electric field gradients around here. Now, if you have negative dP, we would probably trap particles here where the gradient is smallest, and we would trap them here where the gradient is largest under positive dP. And so the overall idea is now, instead of using geometric traps, to use the dP trap. So if you split this up again into 2 half periods, we have the particles actually moving upwards here in this schematics in the first half period, and then once they migrate backwards in the second half period, they would be trapped here due to negative dP instead of a geometric trap. And so I can only show you simulation work at this point. So we tested this with 5 micrometer particles and smaller particles, and actually what you see here is a movie where particles get released in these lines, and we see one period going through. Probably should show this again. So they start here at this line, and we go through one period, so they move upwards and then come back. And so this is the first half period where the smaller force acts, and then when they migrate back, it's the second half period, and we see that they're trapped due to dP. So they end up here. So this is an indication for absolute negative mobility, since the average force points downwards, but they end up upwards. And so now we take smaller particles, and they are released at this line under the same conditions. And you see now what happens with those. So they move upwards, but in the second half period they move downwards, and they actually end up on the other side. Okay? And the reason for this is that just the dielectrophoric force is not big enough in order to trap them, so it doesn't kick in yet. And so this is why these show a normal migration behavior, not affected by dielectrophoresis, where the others do show this. Okay, so they end up down here, versus the other ones ended up here. So we basically transport them into 2 different directions. And that's, if you want, so the best you can get for a separation system if your particles move into different directions. So the previous problem with A&M, and I think this is also why people actually did not follow up with something really useful, is that these A&M velocities were really slow, okay, because they were mainly based on those diffusional aspects, and they were really slow. What we see here now what I want to show you with this graph is that, first of all, if you look at different offsets, we see, again, this typical A&M behavior for the 5 micrometer particles. So negative velocities for positive offset and positive velocities for negative offset. However, if you look at these velocities, they're about 2 orders of magnitude, or not even 3 larger. Okay, so this becomes much more realistic in order to serve as a fractionation or separation mechanism. And then also, what is shown with the red line here is that the smaller particles that we've just seen in the movie, they show a normal behavior. Okay, so they do not show this A&M effect. For a positive offset, they show a positive velocity. And the interesting thing, which I haven't quite understood yet, is the size dependence is inversed. Okay, so now we have the larger particles actually showing A&M, where the smaller particles do not show it, whereas in the geometric A&M center we have seen this reversed. Okay, and this is, let's see, is this important? Yeah, so we can play around with this dielectrophoretic force. So if we make it larger, if we crank it up, if you do, this is wrong. This should be under a larger AC potential actually, right? Yeah, so I mixed up the numbers. Then we see actually that we also can induce an A&M effect for the smaller particles, right? So now we can really play around with this and play around in which particle size we actually move into what direction and how fast. Okay, so this is where I wanted to end, and I hope I could convince you that we can exploit dielectrophoresis for a lot of different things in terms of separating biomolecules, crystals, and particles. And I wanted to thank everybody involved here. So my collaborators here at ASU, previously in Germany at Bielefeld University, the funding sources, since we're also part of XFELS, just recently added on, NSF and NIH, and then the students that did this work. So wanted to point out Bobby, who does all the nanocrystallography related microfluidic work. Jingli, she was very, very tedious in setting up this migration simulation for A&M. And Fernanda and Asuka, they worked on protein dielectrophoresis. Okay. Thank you for your attention.
Alex, in the second part of your talk on dielectrophoresis for proteins, not sorting, I didn't understand the aim of the work. Could you just explain why you're doing it? Is there, for example, some idea that you could control chemical reactions using molecular alignment, or what's the purpose of the work?
Okay, so the purpose is that separations get the better the more parameters you can tune them with, right? And specifically in some applications where we would like to separate proteins, the current methods with which we can separate, they're not sufficient, right? If you look at proteome analysis, for example, right, where you want to separate hundreds or maybe even sometimes thousands of different proteins, so the current methods, they really can sometimes not have the separation power that we need in order to separate.
When they sequence in a gel, they're doing it down to a particular amino acid pair, aren't they?
Not with protein.
It's too slow, is it?
Yeah, but it's not too slow. The resolution is not good enough. What you typically do with proteins, you add another dimension. So you separate them first according to size, for example, and then you separate them according to their isoelectric point. Now, the idea is if you can add dielectrophoresis as another dimension, we would have a larger resolving power for these very, very complex mixtures. So with DNA, you can do this down to base pairs, basically, in sequencing, for example, yeah. Proteins is a different case. Yes.
I might have asked this same question in another seminar, I apologize, but I'm always intrigued when you have these equations that you need the protein conductivity.
Yeah.
How do you model that? What's the meaning of the conductivity of the protein?
Yeah, so the same for anything that I showed probably, right? So,
except plastic maybe, beads, but...
Yeah, so for a biomolecule in a aqueous solution, we usually assume that this conductivity relates to properties of the double layers around it. so the ionic double layer surrounding it, okay? And yeah, that's basically it.
That means that the net charge of the protein matters low. Or doesn't?
Yes, it does. It does in a sense because it determines the properties of the double layer surrounding it, right? And this I have not shown this, but we see this. So the dielectrophoresis is also dependent on what kind of pH, for example, carries out, and it's dependent on what kind of medium conductivity carries out.
So when you said when you go from DNA to proteins, obviously proteins are smaller than the DNA.
Yeah.
But I was also DNA is very charged.
Yes. Yes. So this is why it makes it. This is why it's so tricky with protein. So I think DNA is just a very suitable biomolecules. It's so highly charged, right? And also the higher. Well, so let's say this trapping effect is much easier to achieve with DNA rather than with proteins. Yeah.
In terms of ionic strength of your buffers, the conductivity, the higher conductivity, the worse, right? So
yes, if you have positive dielectrophoresis, yeah.
So in general, you want to use buffers of very low conductivity.
Of low conductivity, yeah. Low ionic strength, which might become problematic in some applications, right? But we're not the only ones. So so there are people that work specifically on that fact when you actually go to higher conductivity. And I would say it becomes even more a problem if you want to deal with cells. Probably, since you want to keep them viable and so on, but with a biomolecule, you certainly do not want to denature it, right?
Right.
But
yeah, that's what I was wondering. Okay.
Any more questions?
I have probably the most stupid question in the room, but how do you avoid these crystals to dissolve? Why is that they don't dissolve at all in the? You have your nanocrystals.
Mm-hmm.
Why don't they dissolve in your?
Why don't they dissolve? Well, first of all, we don't do anything to that solution, so it's right as we get it from them, from the crystallographer. So they do not dissolve.
Is there a buffer being pushed around?
No, they don't. No. Why do they not? Why do they not? It's gentle enough. So, but that's a very important question, right? We want to keep them. We want to keep them intact crystals, right? Yeah. And so this is why we have
separating crystals in a liquid.
Yeah. So maybe, but only maybe, there's a little bit happening, since we really end up with a very tiny size fraction, right? So these 100 nanometers, it actually looks like it's even a little bit better in terms of separation power than it is for the bead. So maybe there's something happening, but they're definitely there. We also checked with techniques that can actually determine whether there's still crystalline material, and those are still crystals. Okay. So, yeah.
So you have electroosmotic flow pulling your fluid through with dielectrophoretic focusing, right?
Yes.
Superimposed. Right?
Yes.
So you've got zero shear flow, which is nice. Well, you have no shear on the crystals, at least while they're in transport.
You mean that's the reason why they're not?
Well, I'm saying at least it's moving like a plug, so that's in your favor.
Yeah. That's right. Yeah, but we were concerned at the beginning whether at these constriction reaches where the large electric field gradients act, whether they would actually not be worth apart or something.
Yeah, I wonder, because if you're actually pulling, if it's all happening at the walls, still you'd have no shear.
Yeah.
It really depends on what's happening when you that could be a break in the model.
Yeah.
That's cool.
Right. Yeah.
What is the effect of electrical field on the proteins? Let's say you separate, but what is the guarantee that they're not going to go under structural change or expression to a different protein?
Okay.
It might be like a general question.
Yeah. So let's say this. In capillary electrophoresis, which is a very well-established standard technique for proteins, DNA or so, there are at least the same order of magnitude of electric fields applied. They always stay intact. Okay, so that's not an issue. Yeah. You certainly have to be aware, we discussed that, because it depends on under what conditions you have them. what kind of buffers and so you use. You do not want to denature the proteins, but they would stay.
So let's say if you want to translate your device into a real application, something similar to what Agile, the chips that you have. So this guarantees like there's no side effect in terms of like, that was my question.
Yeah, I mean, I guess what we have to fight against is temperature effects. If you look for functionality of proteins, you could think of using an affinity assay or something with that. Then you certainly have to look at functionality, whether the proteins stay intact during that. But many electrophoretic applications work under even denaturing conditions, right? So it depends really what you'd like to achieve with that.
And our speaker.